Greetings to everyone. Welcome back once again to this channel on biology. Today we will be discussing a Form 4 topic, that is Chapter 6, entitled Cell Division, and we will continue to discuss some terms such as chromatin, and also we'll go into Chapter 6.2 on the cell cycle. So let's begin. In the last video, we've discussed some important terms, such as somatic cell, gamete, diploid cell, haploid cell, and homologous chromosomes. We also discussed paternal chromosomes and maternal chromosomes. So here's a summary of what we have discussed in the last video. You can screenshot this and have a look at it, or copy it down as a summary. But let's go on to the next part of the lesson. In the last video, some questions were given related to the drawing of cells. So here are the questions again, and we will now discuss the answers. Draw a diploid cell where 2n equals to 2. Okay, the first question. So diploid cell, take note, for diploid cells, there are how many copies for each chromosome? For each type of chromosome? There should be 2 copies of each type of chromosome. Okay, so 2n equals to 2 is very easy. To find out how many different chromosomes you should have in the cell, you just take the 2 here and divide 2 by 2. So you should get 1. There should just be 1 chromosome, but there must be 2 copies of that same chromosome. Okay, so your answer should look like this. Now both chromosomes are the same length because there's only 1 type of chromosome, but there are 2 copies, meaning there's a paternal chromosome, for example the black one, and a maternal chromosome. Okay, so this is a cell which is diploid because the chromosomes, there are 2 copies of each of this chromosome, 2 copies of this chromosome, and it's a diploid cell where the total number of chromosomes is 2. So 2n equals to 2. This is how the cell looks like. Okay, now the second one, 2n equals to 6. Try and draw the cell. If you haven't drawn, just pause the video. It's good to test yourself. Okay, so the easy way of doing it is again, how many different types of chromosomes should I draw of different lengths? Very easy. Just bring the 2 over this side. N equals to how many? 6 divided by 2 equals to 3. You should have 3 different chromosomes of different lengths. Maybe 1 long chromosome, 1 medium-sized chromosome, 1 short chromosome. But because this is a diploid cell, you must have 2 copies of each type of chromosome. Okay? So let's see how you draw it. You can draw first, and you can pause the video and draw, or you can just see the answer here. Okay, this is it. So we have already determined that the number of different types of chromosomes in this cell where 2n equals to 6 is 6 divided by 2 equals to 3, which is also the haploid number. So the haploid number tells you how many different types of chromosomes there are in the cell of different lengths and carrying different genes. So it's 3, so you must draw 1 chromosome that's long, 1 chromosome that's medium size, and 1 chromosome that's short. Okay, and then it's a diploid cell, so there must be 2 copies of each type of chromosome. So you draw 2 of the long ones, long chromosome, 2 of the medium-sized chromosomes, and 2 short chromosomes. Then you can color 1 black and leave 1 white to differentiate between the paternal and the maternal chromosome in each homologous pair. Okay, right, now let's look at this side. Draw a haploid cell where n equals to 1. Haploid, so you can highlight the word haploid. So class, if it's a haploid cell, that means the n number is already talking about the many different types of chromosomes. There's only 1 type of chromosome. Okay, that means you just draw 1 chromosome in the cell. It can be black or white. It can be a paternal or maternal chromosome. Okay, now n equals to 3. It means that there are 3 different sizes of chromosomes, 3 different types of chromosome of different sizes. And is it, are there 2 copies or 1 copy of each chromosome? Only 1, because this is a haploid cell. Haploid cells have only 1 copy of each type of chromosome. Okay? So you just draw 3 chromosomes. Okay? So the n number also tells you how many chromosomes there are that you must draw. If it is 3, it means you draw 3 chromosomes. But the word n here tells you that it is also the total number of chromosomes also. If n equals to 3, there must be just 3 chromosomes. And then because it is n, it means that the 3 chromosomes must be of different sizes, different lengths. Okay? And the n number tells you is a haploid cell. That means there's only 1 copy of each type of chromosome. 1 copy of the long chromosome, 1 copy of the medium-sized chromosome, and 1 copy of the short chromosome. Now n equals to 4 should be very easy for you now. There should be 4 chromosomes of same lengths or different lengths? Different lengths, It should be different lengths. So haploid cells will contain chromosomes of different lengths. You cannot have a haploid cell where there's 1 chromosome which has 2 copies of the same length. That means you cannot have 2 copies of 1 chromosome. That means, let's say this medium-sized chromosome, you have another copy here. You cannot consider that a haploid cell, because in haploid cells, there's only 1 copy of each type of chromosome. So I hope that you have learned something about how to draw diploid and haploid cells from this exercise, okay, which will come in useful to help you to understand better the concept of diploid cell and haploid cells when we talk about mitosis and meiosis. Now for chapter 6.1, we have to learn one more term, and that is chromatin. What is chromatin? Okay, to find out what is chromatin, we must go back to the cell. So in a cell, there is a nucleus. In the nucleus are chromosomes. And each chromosome is made up of a long strand of DNA that's wound around histone proteins. But anyway, we start with the molecule of DNA here. DNA is a double-stranded nucleic acid made up of 2 strands of polynucleotides that are intertwined between each other. And then in the center part of the DNA molecule are the base pairs, nitrogenous base pairs, okay, where the base adenine will bind to thymine and the base guanine will bind to cytosine. Now, what is the basic unit of DNA? Yes, it's called the nucleotide. So here's an example of a nucleotide. Okay, these 3 components, the pink part, the white part, and the yellow part. So can you tell me the components of a nucleotide? Remember the acronym PSP? Please stay back. P is for what? Phosphate group. Okay, the pink part. S is for what? Pentose sugar, the white part. And B is for nitrogenous base. Okay, so in the DNA molecule, there are base pairs that are bound to each other by hydrogen bonds, where A, adenine, will only pair with thymine, and guanine will only pair with cytosine. Okay, so another thing that we have to know is that a segment of DNA forms a gene. A gene is actually a segment of DNA, and there are many genes that are lined up along the molecule of DNA. Okay, now that's DNA. So this very, very long molecule of DNA has to be packed inside a small cell, inside the small nucleus. So to do that, the DNA will combine with histone proteins. All right, it will be wound around the histone proteins. So all these round little structures are the histone proteins. So when DNA winds around them, combines with the histone proteins to form a nucleosome. Here, nucleosome is this structure. Okay, then number 3. Now these are the histone proteins here. Number 3, chromatin is formed from tightly coiled and packed nucleosomes. So all these are nucleosomes and they are packed together and coiled together. So that will form the chromatin. Okay, and from chromatin the chromosome is formed when there is folding and coiling again of the chromatin. So there are many stages of packaging to produce the chromosome, which is actually a coiled up DNA molecule that is really coiled up, okay, where the DNA is coiled around the histone proteins and forms the nucleosome. So there are many nucleosomes, okay, formed and packed tightly inside the chromosome. So chromosome is actually formed from the tightly coiled and packed nucleosomes, which contain the DNA molecule that has wound around the histone proteins. So now we are going to discuss the definition for chromatin. According to our syllabus, chromatin is a chromosome that looks like a long thin thread, long and thin chromosomes. But after that, during the stage of cell division, the chromatin will coil up even further so that it will form the chromosomes that are short and thick, which we can see under the microscope. But before the cell division, during the interphase period, the phase of rest, so-called resting, the nucleus contains the chromatin, which looks like a long thin thread. You cannot see short, thick chromosomes, individual chromosomes. Then, just chromatin. Now, the structure of chromatin is that it is the material that makes up a chromosome, and it consists of a combination of the long DNA molecule and is wound around the proteins called histones. Okay, so DNA is wound around histones to form nucleosomes that looks like beads on a string, like beads being, connected together on a string, so that the long DNA molecules are packed to form more compact, denser structures. Okay, so this is a bit of extra information. Why does DNA have to be wound around the histones? It's to pack them to form more compact, denser structures. Okay, to fit into the cell because the DNA molecule is very long and prevent the strands from becoming entangled. So in the long run, this will protect the DNA structure from harm. That's why it's wound around the histones. However, during cell division, mitosis and meiosis, the chromatin are coiled and folded even further. So we call that condensed. We say that the chromosomes have condensed. And then when it condenses, it will form compact chromosomes that can be seen under the microscope. Okay, so that the chromosomes can be distributed accurately to the daughter cells. Okay, so when they are coiled and folded to form compact chromosomes, it's easier for them to be distributed accurately to the daughter cells without becoming entangled with other things. Now let us study the cell cycle, and the process of mitosis will be studied in the next video. Okay, so this is subtopic 6.2. Describe the phases in the cell cycle. This is our learning outcome. So we must describe first the phases in interphase, which are the G1 phase, S phase, and G2 phase. Okay, where in G1 phase, the cell starts to grow. In S phase, there's synthesis of DNA. That means the DNA is replicated, or the chromosomes are duplicated. In G2 phase, the cell continues to grow bigger. Then comes the second phase. After interphase, we have M phase. In M phase, there are two phases. Mitosis is the division of the nucleus, and cytokinesis is the division of the cytoplasm. At the end of it, two daughter cells are formed. Right, to help you remember the phases in the cell cycle, I have this acronym here, which is very easy to use. Gasing, which is a top. For Malay, gasing means top. Mik. Mik is the name of a person. G for G1 phase. S for S phase, and G for G2 phase. M for mitosis, MI, and CK is actually for cytokinesis. Simple, right? So when you write it, you can put a big G, a small a here, S, big S, I, and G, and then make. Now, what is a cell cycle? So definition for cell cycle is, the cell cycle refers to the sequence of events or an order of events in which a cell undergoes DNA multiplication. That means it will replicate or produce an extra copy of itself, and then there's cell division to produce two daughter cells. So it starts from the time the new cell is formed and then undergoes the DNA replication, and then cell division occurs to produce the two daughter cells. Okay, so the cell cycle can be divided into two main phases, interphase and M phase. Now, interphase consists of three phases. Now, let's look at this diagram. So, generally, there are two phases of the cell cycle, interphase and then M phase. Okay. Now, in interphase, we have the G1 phase, sometimes called the growth phase 1, because the cell grows. S phase, S stands for synthesis of DNA, and G2 phase, sometimes called growth phase 2. Take note that the G1 and G2 phase, the 1 and 2 is a subscript, written as a subscript under at the low part of the G. Okay. Now, after the interphase, where there's growth and synthesis of DNA or replication of DNA, then there is the M phase, which consists of mitosis or nuclear division, and cytokinesis, which is cytoplasmic division. Okay, so mitosis, cytokinesis in the M phase. Now, the M here possibly stands for mitosis. So we are going to discuss all the phases and what happens in them. So this diagram, schematic diagram, shows you the two parts of the cell cycle, which you can copy into your notes to organize your information. So cell cycle is divided into interphase and mitotic phase or M phase. The textbook uses the word M phase. So G1 is the first phase where the cell grows, or there's cell growth, and there's synthesis of proteins and organelles. In S phase, there's DNA or synthesis of DNA. In G2 phase, there's again cell growth, or the cell grows, and there's accumulation of energy to get ready for cell division, which needs a lot of energy. So the cell will accumulate energy in order to carry out that cell division that is coming up. Okay. Then in M phase, we have the process of mitosis, which is nuclear division. There are four phases in mitosis: prophase, metaphase, anaphase, and telophase. The short form is PMAT. Okay, the students like to use this word PMAT, P-M-A-T, PMAT. Okay, for the four phases of mitosis in that order. And then there's cytokinesis, which is the division of the cytoplasm. Here's a basic outline of what happens in the cell cycle. Now, a typical cell cycle will take about 24 hours or 1 day to occur. Okay, with the major part of the time, with the cell being in interphase for the major part of the time. Okay, the M phase is only a very short time, occurs for a short while only, compared to the rest of the cell cycle, compared to the interphase. So it takes about 24 hours for the whole cell cycle to be completed. That is a typical cell cycle, but it may vary for different cells. Okay, now in the first phase, the G1 phase, the cell grows. because it is a tiny cell, it has to grow. As it grows, it will also synthesize new organelles like mitochondria, and new proteins are also synthesized. Then in S phase, the DNA replicates, meaning that from one strand, the DNA will become two strands of DNA that is still attached at the centromere. So this ensures that the DNA will not get scattered and separated from its partner, in the nucleus or in the cytoplasm during the cell division. So our cells are very systematic and organized, and the cells will want to ensure that in the formation of new daughter cells, both daughter cells will get the exact copy of DNA as each other, that the DNA will be distributed equally to each daughter cell. Okay, so for the time being, at this stage, the centromere will make sure that the two DNA strands are together before the process of nuclear division. Then in G2 phase, the cell continues to grow, becomes bigger, right, compared to the original size, and it gathers energy for cell division, because cell division takes up a lot of energy or uses up a lot of energy. So interphase will end after that, after the G2 phase, and the cell will go into M phase. So M phase consists of mitosis and cytokinesis. So the cell will go into this process of mitosis, where the nucleus divides. And there are four phases: P for prophase, M metaphase, A anaphase, and T telophase. The four phases of mitosis. So the nucleus divides, and after that, the cytoplasm will divide in cytokinesis. Finally, two new daughter cells will be formed, and then the cell cycle may repeat, okay, using the new daughter cell to produce, using the new daughter cell to produce more cells. Let us go into the details of each stage. In G1 phase, the cells grow because they are young cells. So each young cell will grow bigger and bigger, and they will produce the cell components such as the organelles like mitochondrion and endoplasmic reticulum inside the cell. Proteins that need to be used in the cell cycle are also synthesized. Okay, and the nucleus looks big. And lastly, the chromosome is in the form of long, thin threads or strands called chromatin. Think of the word thin, long, thin threads, chromatin. In the S phase, a very important event occurs, and that is DNA synthesis. So, to produce two new cells, the new DNA has to be synthesized to be given to the daughter cell, to each daughter cell. So, the DNA in the nucleus is replicated, something like duplicated. Okay, so here you have a strand of a chromosome. It will form two strands. Okay, so the blue one is the new strand because the DNA has replicated. Okay, then later on, these two strands they are identical. These two strands will separate from each other later on in the division of the nucleus. Okay, so each chromosome multiplies into two identical chromosomes called sister chromatids. Now this is one chromosome here. It has multiplied into two identical chromosomes, the blue one and the red one. And they are called sister chromatids. The blue and red one, they are like sisters, okay, and joined together at the centromere. So they are called sister chromatids. If you label two, then you can put their sister chromatids. Don't just label one and say sister chromatid. It cannot be a sister unless the two are side by side, right now. Now then you can say this is a sister of the other one. So label the two sister chromatids. Okay. Both chromatids contain the same copy of the DNA molecule. Okay, to make sure that the same genetic information is passed down to the daughter cells, and that there are no changes in the genetic information. Okay, both chromatids are joined at the centromere. So this is the centromere that joins the sister chromatids together. Okay, this diagram shows the replication of DNA in the S phase. Here we see a chromosome which is made up of a long molecule of DNA. So during the S phase, this chromosome will duplicate and form another strand that's just like it, which is attached to it at the centromere. Okay, so now there are two sister chromatids. They're actually new chromosomes, but we call them sister chromatids because they have not separated yet. Okay, however, if you talk about the number of chromosomes, this is still considered one chromosome, although there are two sister chromatids. Please take note of that. So if the cell has 46 chromosomes, after the replication of DNA, it still has 46 chromosomes. Not 46 times 2, 92 chromosomes. That's wrong. It's still one chromosome, counted as one chromosome like before, but the difference is the DNA mass has doubled. Take note of that. From one strand of DNA, now there are two strands of DNA. So the DNA mass has doubled. But the number of chromosomes remain unchanged. It's not changed. The third phase of interphase is called G2 phase. In this phase, the cell continues to grow. So the size of the cell will continue to enlarge, and it remains active metabolically, meaning that it's still carrying out a lot of chemical reactions in the cell in order to gather energy, because cell division needs a lot of energy. It will also make final arrangements to enter the M phase, which is the next stage of cell division. So after the G2 phase, interphase will end and the cell will go into the M phase. So the M phase is made up of mitosis and cytokinesis. Mitosis involves prophase, metaphase, anaphase, and telophase. So this ends the lesson for this time, and in the next lesson we'll be discussing mitosis. Okay, so thanks for watching. Share, like, and subscribe. I'll see you in the next video. Thank you.